Good afternoon to all the students. So, here we are going to start cell biology. From cell biology, the chapters we need to exercise in NCERT 11 are cell the unit of life, biomolecules, cell cycle and cell division. From these three chapters, you will get 7 to 8 questions for sure in the final NEET examination might be, EAP set might be. That important these chapters are. So, from cell biology, it is very important to build the basic foundation so that when we enter into the new academic year, there will not be any problem, because mostly we will be starting with cell biology itself. So, first if we start cell the unit of life, what are the major contents of cell the unit of life? Let us discuss the major contents of cell the unit of life. What you have to cover up in this chapter? Cell the unit of life. The fundamental insights of this chapter include: first is about cell theory, and basic cell discoveries, different types of cells based on their size and shape, types of cells based on their size and shape, and then complete picture of a prokaryotic cell, and eukaryotic cell. From eukaryotic cell, we have to learn both plant cell and animal cell. Plant cell, it is very important to know about the cell wall structure, composition and different layers of the cell wall. Animal cell, you have to get the complete idea on the fluid mosaic model of cell membrane, fluid mosaic model. Got it, ma? Prokaryotic cell, eukaryotic cell, plant cell, animal cell. In plant cell, you have to be clear on cell wall, their composition and different layers of the cell wall, and animal cell, cell membrane, fluid mosaic model is very important, which is proposed by Singer and Nicholson. And then from a eukaryotic cell, you have to learn about all types of organelles based on amembrane bound, that is non-membrane bound organelle, single membrane bound organelle, double membrane bound organelles. So, in a eukaryotic cell, learn about non-membrane bound, single membrane bound organelles, and double membrane bound organelles. First, non-membrane bound organelles include ribosomes. Ribosomes are the common non-membrane bound organelles which are found both in a prokaryotic cell and eukaryotic cell. In prokaryotic cell, you can see 70 S ribosomes. In eukaryotic cell, you can see 80 S ribosomes. Okay? And then second non-membrane bound organelles will be centrioles. Okay? Second is centrioles. Now, when it comes to single membrane bound organelles, you have to learn about endomembrane system, which is very important. In the every alternate NEET paper there will be one question on endomembrane system. The organelles which are single membrane bound and whose functions are coordinated, those organelles are kept under the endomembrane system. So, here the organelles include first endoplasmic reticulum, both rough endoplasmic reticulum and smooth endoplasmic reticulum. After endoplasmic reticulum it is Golgi bodies, Golgi apparatus or Golgi complex, anything you can say. Then lysosomes and vacuoles. These are the organelles which are included under endomembrane system because their functions are coordinated. And then microbodies also comes under single membrane bound organelles, which are very small minute vesicles which are present in the cytoplasm of the eukaryotic cell. Okay, microbodies. Fine, right ma? Now, double membrane bound organelles. Double membrane bound organelles include nucleus, mitochondria and chloroplast. Nucleus, mitochondria and chloroplast. Though you have already learnt in your earlier classes about all these organelles individually, but here you are going to learn in detail about the components involved in each organelle and functional role of each organelle. Above all this, at the end you are also going to learn in this chapter about cytoskeleton, which includes proteinaceous filamentous structure, includes microbodies, microfilaments and intermediary filaments. Microbodies, microfilaments and intermediary filaments. After this, you will also learn on cilia and flagella. These are the complete fundamental insights of the chapter Cell, the Unit of Life, that you are going to learn in the NCERT 11. Okay. So, first we will begin with cell theory, cell discoveries, different types of cells based on their size and shape, and then complete idea on a prokaryotic cell, and then eukaryotic cell. We are going to learn both plant cell and animal cell. Plant cell we learn about cell wall composition and different layers of cell wall. Animal cell we learn about cell membrane. Thereafter, in a eukaryotic cell, we will exercise all the organelles in this format: non-membrane bound, single membrane bound and double membrane bound organelles. Non-membrane bound organelles primarily includes ribosomes. Ribosomes can be of 70S type and 80S type. 70S type you will see in prokaryotes, 80S type in eukaryote cytoplasm. In both mitochondria and chloroplast, again you will see 70S type of ribosomes only. Okay. And then second non-membrane bound organelles are centrioles. Centrioles you will see only in the animal cell but not in the plant cell. Next comes single membrane bound organelles, which includes endoplasmic reticulum, Golgi bodies, lysosomes, vacuoles. All these organelles, these four organelles put together are kept under endomembrane system. It is because all the organelles here, the functions of each organelle are coordinated. Okay, all the organelles kept in the endomembrane system, their functions are coordinated. Lastly, microbodies, which are small membranous vesicles which are present in the cytoplasm. These are also comes under single membrane bound organelles. Next, double membrane bound organelles include nucleus, mitochondria and chloroplast. Followed by you learn about cytoskeleton, which is proteinaceous filamentous network, which helps both in the intracellular movement and intercellular transport as well. Cytoskeleton is having these bodies, those are microbodies, microfilaments and intermediary filaments. And at last we will learn about cilia and flagella. This is the complete picture of this chapter. So today in this session, from this chapter, what are we going to exercise? We will learn about the basics of cell, the unit of life, which includes cell theory, cell discoveries, and we will learn in detail about a prokaryotic cell. Later we will learn the differences between prokaryotic and eukaryotic cell, and also the differences between a plant cell and animal cell. Let's get started. So yes, this chapter begins with the most noted statement, that is, cell is the basic structural and functional unit of life. If you look into the hierarchy of a living organization, it starts with a single minute atom. Group of atoms put together will form molecules. Group of molecules will form macromolecules. Macromolecules pile up to form an organelle. Group of organelles or set of organelles, when the functions are coordinated, will form up to the cell, and this cell is recognized as the structurally independent, functionally independent structure. Okay, cell is the basic structural and functional unit of life. It is because it is both structurally and functionally independent. Okay, cell is both structurally and functionally independent. Structurally and functionally it is independent. Got it, ma'am? And then group of cells which are of same origin and which have functional similarities. Okay, group of cells which have same origin and perform same function will coordinate and form up to a tissue. Okay, so what is a tissue? Tissue includes group of cells which has common origin, and function, which has common origin and a common function. It can be, those cells can be structurally similar or dissimilar, but if they have same origin and if their function is common, those group of cells will form up a what? Tissue. Okay, structurally they can be similar or dissimilar. Structurally they can be similar or dissimilar. Fine, right? So tissue includes group of cells which has common origin and common function. They can be, those cells, those group of cells can be structurally similar or dissimilar. Then group of tissues will form an organ. Later group of organs set up an organ system, and organ systems will construct an organism. This is the hierarchy of living organization. In that it is noticed that cell is structurally and functionally independent, which is basic structural and functional unit of life. Clear till here, right? Now if we go to the basic cell discoveries, cell is firstly discovered as a dead cell. Okay, firstly a cell is discovered as a dead cell by Robert Hooke. Okay, he has analyzed the cork cells of Quercus huber. Okay, he has analyzed the cork cells of Quercus huber and noticed that these are hollow spaces inside. There is no protoplast inside. These are the hollow spaces. And when he has observed this under a microscope, he said that these hollow spaces cells are like honeycomb structure. Fine, right? Okay. So, dead cells are first discovered by Robert Hooke. They are observed as honeycomb structure under microscope, followed by living cell discovery. Living cell is discovered by Anton van Leeuwenhoek. Anton van Leeuwenhoek. Fine, right? Living cell is discovered by Anton van Leeuwenhoek. He is the father of microscopy. He is the father of microscopy. With the advancement of microscope, the living cells were discovered by Anton van Leeuwenhoek. As they are small unicellular organisms which are observed moving under the microscope, he initially named it as animalcules. Animalcules. Later, it is observed as what? Living cells, but he has initially named it as what? Animalcules. Then, after this basic cell discoveries, one more discovery we need to highlight, that is of Robert Brown. Robert Brown. Robert Brown has discovered nucleus from the root cells of orchid plant. Okay, Robert Hooke has discovered the nucleus from root cells of orchid. Got it? These are the simple basic discoveries that we have to remember at the beginning of the chapter. Dead cells are discovered by Robert Hooke from the plant Quercus tuber. When he has observed it under the microscope, he observed it as what? Honeycomb structure. Second, living cell is discovered by Anton van Leeuwenhoek and he named it in the beginning as animalcules. Then Robert Brown discovered nucleus from root cells of orchid. These are the basic discoveries. Then comes the major concept, that is cell theory, which is very, very important. You have to notice, you have to remember who discovered the cell theory, who proposed cell theory, what are the major points given in the cell theory, who gave modified cell theory, and what are the exceptions of cell theory. All these points are important. Okay. So, first, cell theory is proposed by two scientists. One is German botanist Schlitten, Matthias Schlitten and Theodor Schwann. German botanist Matthias Schlitten, in the year 1838. What is his observation? He has observed all the plants and said that all the plants are composed of cells and products of cells. He proposed one point that all plants are composed of cells. All plants are composed of cells. Next, it is British zoologist named Theodor Schwann. British zoologist named Theodor Schwann in the year 1839. What is his proposition? He observed both plants and animals, said that all the plants and animals are composed of the cells and products of cells. That is the first point. All plants and animals are composed of cells and products of cells. He has also highlighted the point that the limiting boundary of a plant cell is cell wall. Though he is a zoologist, he has set the point that the plant cell's limiting boundary is cell wall, whereas animal cell's limiting boundary is cell membrane. Okay? Plant cell limiting boundary is cell wall over cell membrane, and animal cell limiting boundary includes cell membrane. Okay, in a plant cell above the cell membrane, cell wall is present, is said by whom? Schwann. And animal cell limiting boundary is cell membrane, which is also addressed as plasma membrane. These are the major propositions from the cell theory given by Schleiden and Schwann. In 1838, he proposed that all plants are composed of cells and products of cells. British zoologist Theodore Schwann said that all plants and animals are composed of cells, and plant cells limiting boundary is cell wall, which is over the cell membrane. Animal cells limiting boundary is cell membrane. But both of them failed to explain how new cells are forming. Okay, both the scientists have failed to explain how new cells are formed. How new cells are formed is proposed or said by the scientist Rudolf Virchow. Rudolf Virchow in the year 1855 proposed the concept of omnis cellula e cellulae. Omnis cellula e cellulae. Omnis cellula e cellulae. Okay, which says that new cells are formed from pre-existing cells. Okay. New cells are formed from pre-existing cells. New cells are formed from pre-existing cells. Rudolf Virchow in the year 1855 has proposed the concept of omnis cellula e cellulae, which says that new cells are formed from pre-existing cells. Clear till here? Understood? Can you see this is the diagrammatic representation how new cells are formed from pre-existing cells. This completes the concept of cell theory. Now, cell theory is not applicable to few living entities. Okay, to few cells or few organisms, cell theory is not applicable. To know that to which organisms or which entities cell theory is not applicable, you have to first know that cell theory is applicable to only these types of cells which has a cell membrane. Cell theory is applicable to those cells which has cell membrane, cytoplasm and nucleus at the center. Okay, it should have a cytoplasm and nucleus. Remember students, cytoplasm and nucleus together are addressed as protoplasm. Protoplasm along with cell membrane is known as protoplast. These are the basic terms, but students get confused even before the NEET exam. You have to be clear with it. Okay, cell theory is applicable to those cells which has a cell membrane, nucleus and cytoplasm. Nucleus and cytoplasm are addressed as protoplasm. Protoplasm surrounded by cell membrane known as protoplast. So, you can use protoplast as a synonym of cell again. Okay, only to this type of cells cell theory is applicable. For example, if a cell has more than one nucleus, to such cells the cell theory is not applicable. If a cell doesn't has nucleus, to such cells also cell theory is not applicable. First exception I can tell you is first exception of cell theory. Listen carefully. First exception, we will discuss exceptions of cell theory. Exceptions of cell theory. Got it? First, viruses are addressed as living entities or nucleoprotein particles, or viruses are addressed as living entities or nucleoprotein particles or infectious agents. Viruses are known as living entities or nucleoprotein particles or infectious agents. How a virus will be present? Just a genetic material surrounded by a protein coat. Virus is just a genetic material surrounded by protein coat. So, to viruses cell theory is not applicable. And next example we can take is sieve tube cells and companion cells, sieve tube cells and matured RBC cells, sieve tube cells and matured RBC cells. To these cells also cell theory is not applicable, because sieve tube cells are those cells which are present in phloem tissue, which is a complex tissue. In that sieve tubes are present. These are enucleated, and matured RBC cells doesn't have a nucleus, whereas progenitor cells will have nucleus, but matured RBC cells doesn't have nucleus. These are also enucleated, and as these cells doesn't have nucleus, cell theory is not applicable to these cells. Third is rhizopus. Rhizopus, which belongs to Phycomycetes class of fungi, is cenocytic in condition, cenocytic or multinucleated condition. Cenocytic or multinucleated condition. So, to these cells also cell theory is not applicable. First, viruses. To viruses cell theory is not applicable because just it has a genetic material surrounded by a protein coat. These are addressed as living entities, nucleoprotein particles and infectious agents. Sieve tube cells and matured RBC cells are enucleated. Rhizopus is cenocytic and multinucleated condition. Okay, rhizopus is cenocytic and multinucleated condition. As it has more than a single nucleus, to this also cell theory is not applicable. Got it right? So, these are the exceptions of cell theory which are very important. This completes the whole concept of cell theory. First, you have to know the basic propositions of cell theory. Second, the modified cell theory. Third, exceptions of cell theory. Important. Now, we will directly move on to the differences between prokaryotic and eukaryotic cell, and then we will move on to the differences between plant cell and animal cell as well. We will discuss this in the advanced level. First, we will start with the limiting boundary. Prokaryotic cell is surrounded by 3-layered cell envelope. Prokaryotic cell surrounded by 3-layered cell envelope. Remember, it has 3-layered cell envelope, whereas outermost layer is known as glycocalyx. Outermost layer known as glycocalyx. Second layer is known as cell wall. Third, cell membrane. Glycocalyx, cell wall and cell membrane put together form cell envelope. Glycocalyx, based on the composition, it can be either in the form of glycocalyx, based on the composition, it can be in the form of capsule or It can be in the form of capsule or slime layer. Fine, right? Glycocalyx can be in the form of capsule or slime layer. If the composition is thick, that forms to capsule. If it is loosely arranged, it is slime layer. That is the limiting boundary of a prokaryotic cell. But the limiting boundary of a eukaryotic cell, if it is a plant cell, the outermost boundary will be plant cell, the outermost boundary will be cell wall. Animal cell, outermost boundary will be cell membrane. Got it? So, these are the limiting boundaries. Next, when you get into the cytoplasmic composition, in the cytoplasm of a prokaryotic cell, there are no membrane bound organelles. In a prokaryotic cell, you do not find any membrane bound organelles. Eukaryotic cell will have single membrane bound organelles and double membrane bound organelles also. And then when it comes to genetic material, DNA is mostly double stranded circular DNA, double stranded circular DNA in a prokaryotic cell, double stranded linear DNA in eukaryotic cell, linear DNA. Fine, right? And there are no membrane bound organelles in a prokaryotic cell. Membrane bound organelles do present in the eukaryotic cell. One more specific or characteristic feature of prokaryotic cell are the extensions of cell membrane towards the cytoplasm, either in the form of tubules or in the form of vesicles or in the form of lamellae. It can be in the form of tubules or vesicles or lamellae. Those membranous extensions towards the cytoplasm are known as mesosomes. Mesosomes. These mesosomes, which are the membranous extensions, they will secrete the respiratory enzymes and perform the activity of respiration, perform the activity of secretion. They help in cell wall formation in a prokaryotic cell. They help in the formation of daughter cells, okay, and they also aid in DNA replication. The activities or functions of mesosomes are very important. Mesosomes are what? Membranous extensions of a prokaryotic cell membrane into the cytoplasm. The characteristic features include, first, cell wall formation, DNA replication and respiration, secretion. These are the activities of mesosomes. One more statement you have to remember, as they are performing the activity of respiration in a prokaryotic cell, it is popularly known as mesosomes of a prokaryotic cells are equivalent to mitochondria of a eukaryotic cell. Mesosomes of a prokaryotic cell are equivalent to mitochondria of a eukaryotic cell. These are the basic differences. Now, we will once look into the tabulated version of prokaryotic cell and eukaryotic cell differences. Here we go. First, prokaryotic cell, there is no true nucleus. I said just genetic material is present, but this genetic material is not scattered all through the cytoplasm. It is at a specific location that is addressed as nucleoid. Over the nucleoid, there is no nuclear envelope, or it is not surrounded by any nuclear membrane, okay. So, that is the reason it is said as pseudonucleus. It is not a true nucleus. Whereas in eukaryotic cell, nucleus is covered by a nuclear envelope. Next, DNA structure is circular. DNA is negatively charged. DNA is negatively charged. For DNA condensation to take place, it has to get associated with positively charged histone proteins. But in a prokaryotic cell, histone proteins are absent. In eukaryotic cell, if you see, in eukaryotic cell, the DNA is linear and it is associated with positively charged histone proteins. Histones are composed of the amino acids lysine and arginine. Next, cell size is small in a prokaryotic cell, cell size is large in a eukaryotic cell. Membrane bound organelles are completely absent here. Membrane bound organelles, single membrane, double membrane bound organelles are present in a eukaryotic cell. Cell division is via binary fission, and here it is through mitosis and meiosis. Remember, binary fission is also addressed as amitosis. Amitosis, as there is no involvement of spindle apparatus formation, as there is no involvement or no formation of spindle fibers. Spindle fibers. Ribosomes type is 70S. Here ribosomes type is 80S in the cytoplasm, 70S present in mitochondria and chloroplast. Cell wall composition is peptidoglycan. Here cell wall in a eukaryotic cell, if it is plant cell, it is majorly composed of cellulose. Along with cellulose, it also has hemicellulose and pectin. And chitin is the composition. Chitin is also known as fungal cellulose. Chitin also addressed as what ma? Fungal cellulose. Chitin also addressed as fungal cellulose, which is present in fungi. Cell wall is absent in the animal cells. Next, if you observe the examples of prokaryotic cell, includes bacteria from Eubacteria kingdom and archaea from Archaebacteria kingdom. Okay, and then eukaryotic cells include plant cells, animal cells, fungi and protists. Plasma membrane, it lacks sterols, whereas eukaryotic cell contains sterols. Respiratory enzymes are in cell membrane, which are secreted via mesosomes. These are secreted via mesosomes. And eukaryotic cell, respiration is conducted via organelle mitochondria, which is known as powerhouse of the cell, as this oxidizes the complete food particles and releases out the energy, that is cellular energy, which is ATP. Got it ma? So, these are the clear differences between prokaryotic cell and eukaryotic cell. So, what are the key takeaways of today's session? In the beginning of this class, we have learnt about the fundamental insights of the entire chapter Cell, the Unit of Life, followed by the basic cell discoveries which are to be known, then cell theory whole and soul, okay. Who proposed cell theory, who are the proponents of cell theory, what are the major points to be remembered, and then we have looked into the modified cell theory. Last and very important, exceptions of cell theory, and later we have clearly looked into the differences between prokaryotic cell and eukaryotic cell, both in the diagrammatic version and tabulated form, okay. If these basics are clear to you, when we are moving on to the complete study of a prokaryotic cell, complete study of a eukaryotic cell, fluid mosaic model, composition of cell wall of different eukaryotic cells will not be much tough to you, okay. So, that is all with the today's session. Tomorrow we will move on to the eukaryotic cell in detail and specific. We will analyze both plant cell and animal cell. Thank you.